Negative regulation of B cells. First, I'm going to give you just a small piece of information, of background information. Antibody responses are triggered by binding of antigen to the B cell antigen receptor, which is also known as BCRs. The strength of the resulting signal determines the outcome of the responses, which may vary from induction of tolerance to the antigen to the production of specific high affinity antibodies. So, what is negative regulation? Negative regulation is when a repressive protein binds to an operator to prevent a gene from being expressed. In us, however, it refers to the control mechanisms that have evolved to ensure B cell proliferation is sufficiently slowed down once the B cells have been generated. And once a pathogen have been eliminated, most B cells enter into apoptosis. As you might remember from my last video, I mentioned apoptosis, and apoptosis is defined as programmed cell death. B cells are responsible for the humoral site of immunity because they have the capability of converting itself into plasma cells. Show a little image here. So these are two different images, but we're only focusing on the negative regulation of B cells. So just this part here, negative regulation. CD5B1 cells are known to be unresponsive to B cell receptor mediated growth signals, but instead they undergo apoptosis. However, the B1 cells from Lin kinase deficient mice exhibited an enhanced proliferative response upon B cell receptor mediated cross-linking. And this, according to a study that I found, some reports state that B cell receptor mediated signaling in B1 cells is negatively regulated by signals from CD22, CD5, and CD72 co-receptors, and there are others as well. And the Lin kinase play a major role in tyrosine phosphorylation. Sorry. So, in the absence of CD22, which is here, you can see this co-receptor right here. This is the CD22 expressed on here, and over here is the CD22 expressed on this. In the absence of CD22, it can lower the threshold, the signaling threshold for BCR cross-linking, and it can influence the fate of the B cell. So without the CD22 co-receptor, this can alter the fate of the B cell. A distinct subpopulation of B1 cells was found to express cell surface LY49, which is known as an MHC class 1 binding negative regulatory receptor on natural killer cells. So LY49 is an MHC class 1 binding receptor on natural killer cells. In the addition of the Fab region, which let me see, they have actually the Fab2 region fragments. Here you can see that expressed on both images. In addition of these fragments of anti-LY49 antibodies, particularly block negative signals in B1 cells. So the Fab2 fragments of the anti-LY49 antibodies can block this negative signaling, or block negative signals that comes to the B1 cell. Two co-receptors like the CD5 and the LY49, which are unique to B1 cells. So those are two co-receptors that are very unique to B1 cells, and they play a major role in the regulation of B1 cell activation. And these results actually indicate that B cell receptor mediated signals, also known as BCRs, in B1 cells are strictly and negatively regulated through multiple pathways that are dependent on LYN kinase activity. LYN kinase activity. Sorry. So basically these receptors are needed in the B1 cells, and they can cause a negative reaction, which is regulated through different pathways, and it depends on the LYN kinase activity. Most of these experiments have been conducted on mice and other animals, but I believe that the future is still promising for human development. They develop a lot of ways. They've really kind of expanded it, many different ways in animals such as mice and things like that. And I believe that with more research and more studies, we can definitely find, experiments for human development, which is very important because it can give us answers and results and help us treat things that we have now that we don't have, maybe answers to or something. Like, it can give us answers to these things.